Okay, so if you've been through the reading, which I'm sure you have, you're probably thinking this is going to be a super heavy video. And I'm not saying that it's not going to be a super heavy video. All I'm saying is that I hope you'll be feeling better about it by the time you get to the end of the video, not because it's going to be a whole lot easier, but I think by putting some structure on the content and basically giving you a clear idea of what the objectives are, it won't seem so intimidating as we go through it. And so what we're looking at right now is the first two sections, the first two parts of the cellular respiration or the aerobic metabolism of glucose. The theme that we've developed so far is we've got this general equation, C6H12O6. We're going to be throwing some oxygen at it. Oxygen is like the oxidizing agent that's going to bring us down to get that maximum fall. From the electrons, we got to put 6 there. And then we're going to be ending up with 6 molecules of CO2 and 6 molecules of water, right? And because there's such a huge fall of electrons from the left side of the reaction to the right side of the reaction, in other words, it's exergonics, right? Exergonic reactions are going to be the ones that release energy. The energy that's used is going to be generating ATP, right? So that's the bigger context. We're going to be breaking this down into 4 steps, or 3 stages in the book, okay? And the first 2 steps are glycolysis and the citric acid cycle. Sometimes I might slip and call this the Krebs cycle. You should probably know about that anyways, because you'll be hearing that as well. I mean, there's this thing in the sciences that where we're kind of leaning away from naming things. We used to name things after the person discovered them, but then it quickly became clear that at some proportion of the time, the people that did the discovering, the people that these things were named after, turned out to be real jackasses. For any number of reasons. Maybe they were Nazis, or maybe they were just, child molesters or something like that. Okay, and so as it turns out, we're kind of leaning in the opposite direction. So I don't want to badmouth Krebs. I have no idea. I think Krebs is actually a pretty good dude, but the general theme here is that we are looking at an era in which our cycles are named after what they're doing, as opposed to the person that discovered them. Okay, glycolysis, the citric acid cycle. It also goes by the name TCA, or for tricarboxylic acid cycle. And so, as you move forward in your biology training, you might be hearing a number of names. Okay, and so, in the context of these 2 things, glycolysis and the citric acid cycle, the first 2, we'll be doing the 3rd and the 4th in the next video, okay? The 4 themes that I want to build with you are: 1, what do we mean by a metabolic pathway? Okay? And that's pretty easy. I think you'll find that, okay, I get the idea of what a metabolic pathway is, because we've talked about enzymes and enzyme catalysis. Okay? Second thing, well, what do we mean when we oxidize glucose? Well, that's got specific meaning. We're taking the electrons off of glucose and putting them onto something else. In this case, we're putting them on some type of electron acceptor. Okay? And while that's not entirely easy, if we only think about that one thing, taking the glucose molecule, removing the electrons from that, and putting them on something, okay? Just getting rid of the electrons from glucose, stripping electrons from glucose. That's going to give rise to electrons on some type of, as we said it in the last lecture, some type of intermediate electron carrier. Okay? And so we'll be working on that. That's a concept that, yeah, it's a little heavy, but you can get your mind around it. Not that bad. Next one is actually pretty easy. Dismantling of glucose, which basically means we've got a large molecule, a 6-carbon molecule, and we're going to be chewing it down into 6 little pieces. 6 carbons over here in the form of 6 carbon dioxides. And so by dismantling the glucose on the left side of the reaction, the carbon bits are coming away in 6 little pieces of carbon dioxide. And so that's going to be happening in the process, and that's going to be entirely occurring through the glycolysis and citric acid portion. And we're also going to be accounting for some ATP production. Okay, now, not much. Okay, relatively small numbers of ATPs are going to be coming from this, but we also need to account for the ATPs because, after all, this is kind of like the whole point of undergoing the cellular respiration of glucose. Okay, and so it's a little bit of a pep talk, okay, because I have a slide, and when you look at the slide, it's probably going to be pretty intimidating. But remember, we're starting off with metabolic pathways, and so here is where we learned about how there's a step where you've got reactants and products. I'm just putting reactant and product because sometimes it's only one thing. And then to go to rearrange the bonds, we have to undergo some type of enzyme catalyzed step. Right? We did that a couple of lectures ago. And so basically what we're saying here is that maybe the reactant is a molecule of sucrose, the products here are a molecule of glucose and a molecule of fructose. That requires an enzyme catalysis. The enzyme we talked about was sucrase. But then if the glucose or the fructose were the reactant for the next enzyme catalyzed step, then we could basically see, well, this enzyme is going to be producing a product, and that product is going to be the reactant for the next enzyme catalyzed step. And the product from that is going to be the reactant for the next enzyme catalyzed step. We keep on going through 10 enzymes, 10 steps, 10 enzymes. We'll end up at the end of the process with a final product, which is going to be different, very different from the very initial reactant we're starting off with, right? And so in the next slide, you'll be seeing this thing where I've got arrows, 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 arrows. And that's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. We've got 10 enzyme catalyzed steps that start off with glucose and end up with pyruvate. Actually, 2 molecules of pyruvate because the glucose is being partially dismantled. We're not actually dismantling it completely, but in the process of undergoing glycolysis, the glucose is picked up by the first enzyme. The enzyme converts glucose into this, glucose 6-phosphate. And then the glucose 6-phosphate, this is going to be picked up by the second enzyme. It's going to turn into this, which is glucose 1,6-biphosphate, or something like that. Okay, so at some later point, if for those of you who are going to be biomajors, you'll be learning the entire sequence of steps in glycolysis. But here, I just want you to realize that, yeah, what we talk about as this metabolic pathway is actually a series of enzyme catalyzed steps. Okay. Okay. Now, in the process of glycolysis, we're not going to be seeing a whole lot going on, but there's going to be some things happening. For example, we'll do this in a different color. Okay. The color, I'm not going to try to match the color coding in my next diagram because I don't remember what the colors were. Okay. But here's the thing. Glucose is relatively reduced. Pyruvate is oxidized, right? Which means that electrons have to be coming away from the glucose. We have to have electrons being put onto some type of electron carrier. And like we pointed out in the previous video, we've got this NAD plus, which is an electron acceptor. And once you put electrons on it, it becomes NADH, right? And so in other words, we're making an electron carrier which is that kind of intermediate step in the harnessing of the energy that's coming from the oxidation of glucose. And so, yeah, electron carriers are receiving the electrons. We're not really dismantling it too much. We're basically taking glucose and breaking it into two bits, two parts, two large parts, two 3-carbon bits, pyruvates. And we're also, and this is kind of a different thing, okay? So we've accounted for a little bit of the oxidation of glucose. We're also doing this other bit, which is to produce ATP, right? So in addition to making NADH, we're also going to be generating ATP from ADP plus phosphate. Okay. And we're actually going to be doing that 2 times. Okay, so we'll be making 2 molecules of ATP. We'll be making 2 molecules of NADH because we're taking electrons, enough electrons to reduce 2 molecules of NADH. Right? And so, yeah, I mean, there's a lot going on, but think of it as 4 things, actually a lot going on. There are only 3 things. We'll be looking at the metabolic pathway of glycolysis. Remember that happens in the cytosol. We'll be looking at the metabolic pathway of the citric acid cycle. And in each one of those, we should be able to account for a little bit of oxidation, a little bit of dismantling, some ATP production. Okay, ready? Okay, now here's the diagram. And so what I've done here is to show you, well, this is a metabolic pathway that we just described. This is the metabolic pathway of glycolysis. Okay. Lysis means splitting. Glyco means sugar. So glycolysis is the splitting of the sugar. And we're actually breaking a sugar molecule, a 6-carbon glucose, into 2 3-carbon molecules of pyruvate. Okay. And as I said before, we're making 2 molecules of NADH. This is the part where we're actually seeing the oxidation of glucose, taking electrons off. In this case, we're taking 4 electrons off because each NAD plus is receiving 2 electrons. Okay? 1 electron is going to neutralize the plus charge. The second electron is going to be met, and basically it's going to carry with it a hydrogen ion. So we end up with NADH. And that's going to be one of the things. And then you look also, we're talking about some oxidation of glucose. We're seeing not a whole lot of dismantling glucose. Okay, maybe not so much of this, but we are seeing ATP production, right? We're getting 2 molecules of ATP being produced from ADP and phosphate. Okay. This is what happens in the cytosol. Glucose molecule gets taken in by a cell. It gets attached by the first enzyme in glycolysis. By the time it gets to the end, you've got 2 pyruvates that are ready to make the migration into the inner matrix of the mitochondrion. A mitochondrion is singular for mitochondria, in case you're wondering. Okay. And as this is happening, basically, as the pyruvates are being imported into the inner matrix of the mitochondrion from glycolysis, pyruvates are produced by glycolysis out in the cytosol, and then it gets transported from the cytosol over into the inner matrix of the mitochondria. What happens is that the pyruvates are converted, okay, into 2 acetyl groups. Okay. Acetyl groups are also going to be oxidized relative to pyruvate. In other words, pyruvate relative to the acetyl groups are reduced. Acetyl groups relative to pyruvate are oxidized, and the electrons are coming off. 4 more electrons are coming off. We're making 2 more molecules of NADH. Okay? And here's where we're beginning our first, final dismantling of glucose, because ultimately we're going to be making molecules of CO2. And so the pyruvates are 3 carbons, acetyl groups are 2 carbons, and so the last carbon is going to be coming off as carboxyl. It's called a decarboxylation, and that one carboxyl group is going to be coming off in the form of the CO2 from each of the pyruvates. Okay? And so, yeah, we can actually see the dismantling of the glucose right there, right? We're taking glucose apart. It's red. We're taking glucose apart and releasing CO2, right? So here are 2 of the molecules of CO2. Ultimately, we're going to account for 6 molecules of CO2 being released, because that's what we have here, right? We expect to have 6 molecules of CO2, and 2 of them are coming off in this transition between pyruvate and the acetyl group, right? The other 4 are going to be coming off in the citric acid cycle, right? And so between the citric acid cycles, 4 CO2, and the 2 CO2 that are happening before, we're accounting for the final dismantling of glucose and the release of the CO2. Okay. Now, the other thing, again, going back to the oxidation of glucose, we're oxidizing the glucose further in the citric acid cycle. Let me pick another color for this. So let's do this in yellow. This here, undo. This is the citric acid cycle. And what's going on, it's another 10-step enzyme catalyzed pathway. The acetyl group is going to be joining with something to make citric acid. So in other words, citric acid is the intermediate. It's right there. The citric acid is picked up by the next enzyme, and it's going to be producing a product there that's going to become the reactant for the next. This happens. We keep on going through, and finally we end up right back where we start. And at that point, we'll be putting on another acetyl group, right? This is happening. The cyclical enzyme pathway is happening inside of the mitochondrion, and along the way, we're basically seeing the release of 4 more molecules of CO2 from the 2 acetyl groups that are going in, right? So yeah, we're accounting for the final dismantling of glucose. We're getting a substantial amount of oxidation as well, because we're also seeing more NADHs forming, because electrons are falling off of these acetyl groups, getting put onto NAD+ to make NADH, and we have the second kind of electron carrier, FAD. It's basically a variation on NAD+. Don't worry about FAD. The concept here, again, is that we're putting electrons on carriers, on electron carriers, which allow us to capture the energy from the breakdown of glucose, the complete oxidation of glucose. Okay. Last thing is that we have ATP production. ATP production is here. And so remember that we had 2 molecules of ATP being produced in glycolysis. We have 2 more molecules of ATP being produced during the citric acid cycle. And so we get a total yield of 4 ATPs, which is actually not very much. 4 ATPs produced out of roughly 36 ATPs total that will be produced with the aerobic metabolism of glucose, which basically tells us that the remainder, the other 32 ATPs that are produced, are going to have to happen in that last part of our cellular respiration process, in the electron transport system combined with the chemiosmosis, which is the process we're identifying as oxidative phosphorylation. That's the next video. Okay, so, focus right now on glucose, citric acid cycle. Focus right now on the idea that these are metabolic pathways, 2 metabolic pathways, which we've got enzymes picking up a substrate, which is a reactant, giving rise to a product, releasing the product, and then that product is the substrate, the reactant for the next enzyme. It happens in the cytosol, giving rise to the glycolytic pathway, happens in the inner matrix of the mitochondrion, which is the citric acid cycle. Okay. Second thing, so the other thing that's happening is that we're oxidizing glucose, and we're doing so completely. We're taking all the electrons off of glucose and putting them onto some type of carrier, right? NADH, and if you like, FADH too. Okay. Further, we're dismantling the glucose. We're not being shy about this. We're taking the glucose and breaking it down all the way to CO2, right? And so all the CO2 is accounted for, and all the carbons are accounted for at this point. Now all we have to do is to account for the ATP produced, the other 32 molecules of ATP, because we're accounting for 4 out of the total of 36. Good? Okay, so now let's take a look at the textbook and see how the textbook is defining this. Okay, so chapter 6, part 7, glycolysis is harvesting chemical energy by oxidizing glucose to pyruvate. You know all about that at this point, right? We're taking glucose, we're taking the electrons off of glucose and putting the electrons onto NAD+. This is our electron acceptor. And then we make the electron carrier, NADH. And we're also making 2 molecules of ATP. And that's exactly what we've been talking about so far. In other words, this is more or less identical to what we've been talking about. It also kind of identifies how the ATP is being produced. Don't worry about that. All we care about right now is that ATP is being produced. Moving on to the next portion, reactions in glycolysis here we're actually talking about what we mean by an enzyme catalyzed step, right? By what we mean by metabolic pathway involving multiple enzyme catalyzed step. So annotation function. This glucose here is the reactant for the first enzyme, and the glucose phosphate is the product of the first enzyme. And then the glucose phosphate is the reactant of the second step, and the fructose phosphate is the product. And then the fructose, basically we keep on doing this. And as we go step by step, by the time we get to the bottom of the picture, oops, let me here are my drawings. At the bottom of the picture, we've got pyruvate. Okay? And if you notice, pyruvate is a 3-carbon molecule. That's all well and good. There's a little bit more detail, like the fact that in the first part of glycolysis, we have to invest 2 molecules of ATP, and we get those molecules of ATP back in the second half, but we get 2 more. In other words, we put in 2, we get back 4, and therefore there's a net yield of 2. Okay? Nice little thing to know about. Not as important as the things that I've been emphasizing with this video. Okay, now chapter 6, part 9, citric acid cycle. More or less exactly what we've been talking about so far. We're going to be starting with moving the pyruvate into the inner matrix of the mitochondrion, and in the process we'll be making 1 molecule of NADH. For each pyruvate, we'll be releasing 1 CO2. In other words, we're dismantling the carbon. We are oxidizing the carbon, oxidizing the glucose, still taking the electrons and putting them onto NAD. Right? And when we get to the citric acid cycle, we're making more molecules of NADH, FADH2. These are electron carriers, and we're producing a molecule of ATP. Right? Now the numbers here are a little bit different from what you have on your in your notes so far, because because this is only 1 turn of the citric acid cycle. There are 2 pyruvates that are produced early, right? There are 2 pyruvates that are produced from glycolysis. And so we actually have to go through this twice. And that's how we end up with twice the number of molecules of CO2, twice the number of ATP, twice the number of FADH2, as well as NADH. I think I made a mistake. Previous drawing, I think I gave you a total of 4 NADHs. I guess it should have been 6, right? So, okay. And again, moving on to the next step, just more detail. But again, the detail, not crucial at all. I mean, I really don't care about the details here. What is alpha-ketoglutarate? Can you even pronounce alpha-ketoglutarate, okay? And then there's succinate, fumarate, malate, oxaloacetic acid, which is the same as oxaloacetate. And so, we go from one molecule to the next. We make, we do what we need to do. Let's just, I think the other share that I had, the whiteboard, this diagram is scary enough, right? This is concerning enough because it does provide an adequate amount of detail, and we're understanding things in terms of, one, we're oxidizing glucose. Yeah, I understand what's happening there. Electrons are being removed. What happens to the electrons? Well, they're getting put onto NADHs and FADH2s. This is a place where I had it should be a 6, right? We're dismantling the glucose. What is happening to glucose? Well, remember, glucose is 6 carbons. We're getting 1, 2, 3, 4, 5, 6. We're getting 6 carbons in the process. They're being released in the form of carbon dioxide. And we're also accounting for some ATP production. 2 of them are being netted in glycolysis, and the other 2 are coming from the citric acid cycle. Okay? Yeah. You might want to take another look at the chapters. I know you probably read through the chapters and thought, well, what is all this about? Okay? I hope that now you should be able to go back to the chapters and make a lot more sense out of them. Okay? If not, bring your questions to our meeting and we'll talk about them then. Bye.